This is a production of Cornell University.
Okay, hello everyone. I'm Jacob Toth. I work in Larry Smart's lab in Cornell Agritech up in Geneva. And today I'll be talking about accumulation of cannabinoids under various stresses in high CBD hemp, presenting some results from what we've been calling the 2019 Hemp Stress trial. So hemp is a cannabis sativa plant with less than 0.3% total THC. And the major market for hemp right now is cannabidiol or CBD, which is already $1 billion market and is expected to grow exponentially over the next few years. CBD is isolated from hemp inflorescence, and it reaches its highest concentration in mature unpollinated female bracts. One of the things that a lot of people are concerned about when growing hemp for CBD production is the production of the psychoactive tetrahydrocannabinol or THC, which is what distinguishes hemp from marijuana. And there's an arbitrary threshold of 0.3%. So knowing what results in higher or lower production of THC is going to be very important for growers who want to stay under that limit. And if you look at sort of what's popularized in the media, or at least what's popular when these articles were written, it appears that hemp stress is expected to have a significant effect on THC. So if you look at the hemp and the marijuana blogs, for instance here for tuna hemp, suggests that growers need to be aware that plant stresses can result in THC spikes. RX Leaf suggests that any plant stress can tip it over 0.3. Even cooperative extension says that the main thing that causes THC spikes are stressors. And the New York Times mentions a quirk in the chemistry of hemp that can cause them to overproduce THC when stressed. So while a lot of people think that hemp stress is going to be very important, it made it all the way to the New York Times, there's very little evidence in the literature that links hemp stress and THC production. Now, part of that's because hemp has only recently been legalized for production in many states. So what we wanted to do was look explicitly and see if these sort of popular sources have any truth to them that hemp stress is going to result in higher THC. So just looking at previous work going into this, we didn't expect that hemp stress would have much effect on THC spikes. In 2018, we found that there was a fairly consistent CBD to THC ratio among genetically similar plants. We ran a little bit of a natural experiment as well. We grew 2 replicated fields, one in Ithaca and one in Geneva. And one of our fields was severely flooded. And you can see here that plant is pretty stressed. But we didn't see any effect on the CBD to THC ratio, and we didn't see any THC spikes within genetically similar plants. Now, this isn't a perfect experiment. There could have been other factors at play, and there may have even been some selection bias as the plants that were really stressed here just died. So what we wanted to do was run a randomized complete block design experiment to see if stress affects the THC levels and if stress affects this CBD to THC ratio. So to do this, we had 3 cultivars in 4 reps with 6 total treatments, which I'll discuss in a minute. And we took shoot tip samples every week for 4 weeks, and then using HPLC looked at the cannabinoids that were produced. So we had 6 treatments. We had our control plants, which were grown under best practices under black plastic. We had powdery mildew, which is a fungal pathogen, which we thought could induce a response. Rather surprisingly, we're not 100% sure on the evolutionary purpose of cannabinoids. It's been suggested that they might be a factor in pest response or pathogen response, or maybe even UV response. So we included powdery mildew because we thought it might induce more cannabinoids there. Mechanical damage is something that people are concerned about. It may hail, there may be some wind damage or potential herbivory. So we included that as a potential stress, and to generate that stress, we damaged the plants with a grass trimmer. We had a flooding treatment which saturated the soil regularly, and this was largely to repeat that 2018 finding in the controlled experiment. We also included 2 chemical treatments, Ethaphon, which was found to increase total cannabinoids in a greenhouse study of marijuana, and a herbicide at a low concentration to emulate herbicide drift. So that's what you see here. These plants have been sprayed with 0.5% Roundup, and they're pretty stressed. So this is just the control. This is best practices, and this matches a lot of the other data that we've been seeing. The CBD increases over time, and it's somewhat cultivar dependent. So it started at week 0 before application of these stresses. All of the cultivars are around 3% total CBD. And then at the end of the experiment, the CBD has either leveled out in that one case, or is around 10%, which is usually where growers aim to take the crop. So that's CBD. And if we look at THC, the graph is actually very similar. So if you look at this total potential CBD in this little inset and the total potential CBD in this larger graph, they look very similar ratio of CBD to THC over time. What's also very noteworthy looking at this is that these 2 cultivars, the blue and the red, they're significantly over that 0.3% THC, even under best practices, as unstressed as we could get them. So if there's a very similar ratio of CBD to THC, we'd expect that even without any additional stresses or anything else, at around 7 to 8% CBD, we'd be getting non-compliant levels of THC, and under some circumstances, the crop might have to be completely discarded. So that's in the control. We wanted to see if there was any effect of any of these stresses on either the ratio or the total amount of cannabinoids. And largely, there was very small effect of stress. So if you look at this total potential CBD, you can see that most of these treatments cluster with the control except for herbicide, which is generally a little lower. And given how stressed those plants are, that's not altogether surprising. The plants were very significantly stressed. They were fairly close to death. We had trouble finding green tissue to sample. If you look at the total potential THC, it's a very similar story. It looks like the ratios are about the same throughout. You see lower herbicide, and that's, we think, largely because of just sort of general stress. The plants are nearly dead. We also did not see any huge spikes in THC, which is sometimes what has been reported. But when we look just at the harvest time here and look at it statistically, we can see this total THC. The only significant difference compared to the control is that the herbicide has lower total THC. So it appears that stress does not cause THC to spike. What's more, if we look at this ratio of total CBD over total THC, this ratio remains the same in all comparisons at harvest. So to explain why that happens, I'm going to talk a little bit about the production of cannabinoids. So first off, the first cannabinoid that gets synthesized is CVGA, and the amount of CVGA that gets synthesized is going to be a function of a few things like trichome density, production of this olipholic acid precursor, activity of this GOT enzyme, a whole bunch of things like that. And a lot of these things apparently have a strong genetic base, is what we've seen. These stresses didn't affect them too much, but can be slowed down by nearly killing the plant with herbicide. So what happens in hemp with CBDA synthase is the CVGA gets converted to CBDA, which can then be converted to CBD. And in marijuana, you have THCA synthase, which converts to CBGA to THCA, which then gets converted to THC. So as I pointed out a few times, these hemp plants, which should only have this CBDA synthase, produce a fairly consistent ratio of CBDA to THCA. And as it turns out, if you isolate that CBDA synthase or express it in a yeast system and incubate it with the CVGA, you get a small amount of production of this THC as well. And what's very encouraging is that the ratio that you see in vitro is very similar to the ratio that we see in vivo. So these hemp plants that we're growing, that THCA is the result of promiscuous activity of the CVGA synthase enzyme. And none of these stresses apparently affect that promiscuous activity of the enzyme. So THC spikes are a real thing. It's just a lot of people have been attributing it to stress, while we could not see any spikes due to stress. And to summarize why we think that is, just in a few words, is because those plants have active THCA synthase enzyme in them. So cannabinoid type can be accurately modeled by a single locus called the B locus. It comes in two major flavors, the BT, which has the active THCA synthase, and the BD, which has active CBDA synthase. And since hemp is diploid, you can get three chemotypes, as they're called, BT, which produces mainly THC, BT, which produces significant amounts of both, and then chemotype 3, BD, which is what you would think most CBD hemp should be, but a lot of the commercially available cultivars are not solely chemotype 3. A lot of them have significant numbers of chemotype 2, and some even have some chemotype 1. So here are 4 different cultivars that we grew. This first one here that's all blue is all chemotype 3. The next one has a little bit of chemotype 2, and this one in the bottom corner was in fact 2/3 chemotype 2. So unsurprisingly, it produced a lot of THC. Now, if you were just randomly sampling, you might randomly pick a plant that's blue, chemotype 3, and then, you know, a week later you pick one that's orange, chemotype 2, and blame it on the stress, when in fact, it was simply a genetic phenomenon. It was just a different chemotype due to a different gene at that B locus. So in summary, stress has the potential to alter cannabinoid content. For instance, if you nearly kill the plant with herbicide, cannabinoids will be lower, but the effect was generally fairly minor. Very importantly, the stress does not affect the CBD to THC ratio at harvest. And we think that the spikes that people have been reporting are likely due to sampling different chemotypes, as we're seeing that chemotypes segregation is very common in many cultivars. Another important result is that high CBD plants are almost certain to test above 0.3% THC when grown to a high CBD percentage. So what we've been advising growers to do is to ensure that they're growing seeds from a reputable source that ideally don't have any of that active THCA synthase enzyme, and also to not get too greedy, to test and harvest before the plants get too high of level of CBD, so that it can remain below that threshold of THC. So for acknowledgements, I'd like to thank other members of the Larry Smart Lab, particularly George Stack and Craig Carlson. I'd like to thank Chris Smart's lab and Ali Kalla for their work with powdery mildew, and the Rose Lab for working with the HPLC to determine these cannabinoids. This work was largely funded by New York State Department of Ag and Markets and Pixis International. And with that, if I have any time left, I'd be happy to take some questions.
Great. Thank you for that really thorough talk, Jacob. If anybody has any questions, they can either unmute or put them in the chat, and make sure to fill out that feedback form. I put the link in the chat as well.
Hey Jacob, this is Travis. I have a question for you. What is the amount of heat that's needed to convert the precursor to CBD into CBD?
Right. So CBDA and THCA get converted into the active forms of CBD and THC. And there's no, if you look at the decarboxylation profiles, they're temperature dependent. So we've had samples that have been left at room temperature for a year, and they end up being mostly decarboxylated. But one of the reasons why marijuana is smoked is because at high temperatures, it gets decarboxylated very quickly. So there's a time temperature dependence, and they are different for different cannabinoids, this decarboxylation, and that's an area of active research.
Okay, thanks.
I think there's one more question in the chat before we move on. Faith asks, have you considered including mechanical stress for another experiment?
So we were hoping that our mechanical damaging the plant would sort of encompass any sort of major mechanical stress. It might be something different, given that we saw very clear no results. I don't think we have immediate plans to repeat work like this. We have a nice theory that it's just that one enzyme, and we don't foresee it being affected by mechanical stress, and we didn't see an effect of our physical damage. So it would be another stress that could potentially be included, but we don't really have any immediate plans to test that.
Awesome. Thank you again, Jacob. We can all give him the clap emojis. And I think Anna's going to introduce our next speaker.
This has been a production of Cornell University, on the web at cornell.edu.